So this study really dives into how specific intestinal microbes can, you know, enhance muscle strength, right? It's fascinating. They transplanted human gut microbiota into mice and then saw, well, variable effects on the mice's muscle performance, measured by tests like rotor rod and wire suspension.
That's exciting. And it's not just random improvements. They actually identified distinct groups. Some mice got stronger, others didn't, based on differences in their gut microbial communities.
Exactly. What's really interesting is that they compared fecal samples with the actual gastrointestinal content and discovered that the GI tract had a much richer and diverse microbial profile than just the feces.
Yeah, that's a key point, right? Right, and with that deeper dive, they used 16S rRNA sequencing to tease out these differences. They found increased species richness post-transplant, even though the evenness stayed about the same across samples.
And then, after all that data crunching, they homed in on two critical bacterial species, Lactobacillus johnsonii and Limosilactobacillus reuteri. These two were consistently enriched in the groups showing enhanced muscle strength. It's like nature's own power-up for muscles.
Yeah, it's pretty remarkable. They even went a step further and administered these strains to aged mice, and guess what? Their muscle strength improved significantly. The rotor rod performance and wire test numbers shot up, and muscle mass increased.
Yes, and when we talk about mechanisms, they observed upregulations of molecular markers like follistatin and IGF-1, which are well known for promoting muscle growth. So it really forms a bridge between the gut microbiome and muscle development.
That's a pretty solid link. Although I wonder, don't you think it's a bit early to say these are a definitive treatment for muscle atrophy in humans, given that the study was in mice?
Absolutely, that's a fair point. The experiments were in a controlled murine model, so while the findings are promising, we need further human or ex vivo validations to truly translate these findings into clinical
Right.
applications.
More studies will definitely be needed. Another cool aspect was how they analyzed not just simple abundance, but used differential abundance analysis to identify key strains. The fact that these microbes, especially in the GI samples, correlated linearly with muscle strength improvements suggests a causal role.
Yeah, and it really underscores the complexity of the gut-muscle axis. Their use of multiple tests, diversity measures, phylogenetic trees, and co-occurrence networks provided robust evidence that the microbiome structure, especially in the GI tract, is pivotal for muscle function.
Solid multi-angle approach. And then, when they tested combining L. johnsonii and L. reuteri, the synergy was clear. The combined treatment produced a significantly higher muscle mass and performance, with markers skyrocketing compared to individual administration.
Exactly. It's like they're not just adding up their effects, they're amplifying each other's impact. That synergy is what really might pave the way for novel probiotic therapies targeting muscle-wasting conditions.
Indeed, future treatments could be like muscle cocktails tailored from your gut microbes. Overall, this study gives us a lot to chew on about the potential of microbiome-based interventions, though we should be cautious about overgeneralizing from mice to humans.
Yep, agreed. It's a groundbreaking step that opens up exciting prospects for understanding how our gut can influence muscle strength and ultimately health.
Absolutely, very inspiring work.